Okay. So I have network planning for today. I think at some point I'll just start a Gemini discussion and ask like what I can do for that. That said, I think I still want to continue what we've been working on. Which is making more sense of the chemicals that are involved in our focus and attention and motivation. We've been reading this guy. We went through norepinephrine, acetylcholine, and then dopamine. And we're on dopamine right now. We're on liking versus wanting. So we saw these guys. We know we've got Go system, regulating incentive salience, desire, and the psychological feeling of wanting. We discussed here that this touches upon this gets a lot of stuff into motion, including norepinephrine and acetylcholine. This also involves releasing opioids, which give us like a feeling, like a positive feeling. It amps up our system and it gives us a positive feeling, which makes us feel as though there's something good pulling us in a direction. So what do we do? Where do we get to this? We talked about the interpreter theory. Insula. Dopamine creates motor tension, the spring. Opioids provide a sensory preview. The insula feels the body revving up, and then the insula basically interprets what's going on based on everything that's happening. Cortex concludes, I'm choosing to pursue this because I want it. So this is how we end up with this idea that we want something. There's kind of a paradox of like, how do you know that you want this thing? Did you decide that you want it? So you obviously you could say I decide now that I want it, but like what gives you what, like, did you make the decision to even get to the point where you feel like you want it? And so, all right, let's just continue. Liking versus wanting, the dopamine opioid dissociation. A critical distinction in the neurobiology of reward is the separation of liking, hedonic impact, from wanting, incentive salience. These two psychological components are mediated by dissociable chemical systems. So does this mean just different? Or separable? Wanting, dopamine. Microinjection of dopamine agonists or amphetamine into the nucleus accumbens dramatically increases the effort an animal will expend to obtain a reward and increases the consumption of the reward. However, it does not increase the orofacial expressions of pleasure, for example, tongue protrusions. This is the state of craving without satisfaction. And then there's liking, opioids, endocannabinoids. The sensation of pleasure is mediated by small hedonic hotspots, cubic millimeter sized zones in the rostrodorsal quadrant of the NACC shells. Stimulation of mu opioid or delta mu opioid or delta opioid receptors in these hotspots enhance liking reactions. Dopamine alone cannot trigger this hedonic response. In a healthy flow state, these systems work in concert. Dopamine drives the pursuit, and opioids provide the reinforcement upon success, which in turn reinforces the dopaminergic learning signal. Addiction or burnout can occur when these systems become uncoupled. High dopamine drive with diminished opioid feedback leads to compulsive behavior without fulfillment. So I'm still trying to understand and grasp how these work together in cases where it doesn't seem like a reward has been met, but chances are the brain feels as though a reward's been met. Like some sort of progress towards a goal probably results in opioid release, which makes it feel as though your pursuit is good in some sense. I'm just trying to figure out how we, like, why do we actually feel good about dopamine after the fact, except for that final release. All right, reward prediction error, the learning signal. Dopamine neurons do not fire simply because a reward is present. They fire based on information. They encode the discrepancy between the expected reward and the actual reward. So, better than expected, a phasic burst of dopamine. This strengthens the synaptic connections, long-term potentiation, that led to this action, reinforcing the behavior. Negative RPE, worse than expected, a dip or pause in baseline dopamine firing. This acts as a punishment signal, weakening the synaptic connections and discouraging the behavior. So, what did I want to know about this? So long-term potentiation clues us in that it's not just that neurons firing leads to strengthening. It's that neurons firing like leads to a window of time in which this phasic burst of dopamine could affect the strengthening of that. All right, so let's actually figure that out. Can you help me understand long-term potentiation? What allows neuron firings to be open for future strengthening if phasic dopamine occurs? I don't know if that's the right way to say that. Yo, double gem finding gems. A little cryptic. Double gem finding gems. What gems have we been finding? Oh, Demel Gemini. Hell yeah. I'm locked and loaded. Yep, gotta get the double Gemini going. It's like I'm still in disbelief at the opportunity of being able to ask questions and then ask follow-up questions to learn stuff like this. Like, this is insane what I'm doing right now. Like, I'd have to read books and then I wouldn't even be able to answer questions unless I found what I'm looking for in another page of the book at another section. Maybe I could look it up on Google. Maybe that works, but you know, in these very niche topics, those types of questions are very difficult to just like get to. I'd have to be looking at papers maybe. All right, long-term potentiation is the biological process of memory formation. It is how a fleeting electrical spark transforms into a physical structural bridge that lasts for years. To answer your specific question about openness, the mechanism that keeps a synapse open for future strengthening is called synaptic tagging and capture. This hypothesis solves a major engineering problem. The nucleus, where proteins are made, is far away from the synapse where the memory happens. How does the nucleus know which of the 10,000 synapses to send reinforcement supplies to? Ah. Okay. Here is the step-by-step breakdown of how a synapse is tagged and how dopamine seals the deal. 2 stages of long-term potentiation. Between the draft and the final copy. So early long-term potentiation. This is the draft. It happens instantly when neurons fire together. Glutamate. Glutamating. The synapse gets stronger for about 1 to 2 hours. It uses existing proteins floating nearby. Crucially, if nothing else happens, this strength fades away. But late long-term potentiation, this is the final copy. It requires protein synthesis, new construction materials. This lasts for days, months, or years. Okay. The tag, opening the window. When a specific synapse fires strongly, so glutamate, NMDA activation, 2 things happen locally at that specific dendrite. This is the input of a neuron, effectively. Immediate boost. AMPA receptors are, I don't know what AMPA means, but nor do I know what AMDA means. NMDA. AMPA receptors are pushed to the surface. The synapse works better temporarily. Okay. And the molecule tag. The synapse gets a chemical flag, like a modification of the actin cytoskeleton, or a specific kinase of like CAMK2. All right. So what's the takeaway here? Receptors are more not excitable, but they're more eager to they're more sensitive, let's just say. And then the synapse actually tags. What's it tag? Is the tag just kind of sitting there now? This tag is the openness. It marks that specific doorway as ready for construction. The tag is temporary. It decays in about 60 to 90 minutes. While the tag is active, the synapse is waiting for plasticity-related proteins. 60 to 90 minutes. So you have to succeed in what you were trying to do within 60 to 90 minutes. Now, here's another question. This is going to get pretty nasty, though. Could we not set this up and then do something else that is high phasic dopamine in order to strengthen the previous connections? So that's the crazy question to ask. Okay, if synapses are open for a significant amount of time, open for okay, 60 to 90 minutes. Is it possible to game the system such that you exercise those neurons, synapses, and then immediately do something else that is highly stimulating? Like choose any high dopamine activity. And perhaps you'll need to ensure that those same synapses are covered. So you would need to take brief moments to think about the activity activity/information you were previously doing, which you want to strengthen. All right, that's gonna be the first crazy idea here. All right, the dopamine signal, ordering the supplies. This is where the phasic dopamine comes in. While the synapse sets the tag locally, dopamine acts globally, or at least broadly, on the cell body. Okay. Oh, so this is tricky. You mentioned that phasic dopamine acts globally, so maybe this would hit the previous synapses. Otherwise, perhaps you'll need to ensure that those so you would need to take brief moments to think about the okay. A reward or novel event releases dopamine. Dopamine hits the receptors on the neuron. This triggers a chemical cascade that travels to the nucleus. The nucleus starts printing new proteins. All right, the capture. This is the magic of the system. The nucleus releases these new proteins blindly. It doesn't know where to send them, so it sends them everywhere down the dendrites. Okay, so it's sending them backwards, effectively. Backwards in the sense that this is where the signals are coming in. It's sending them back down to strengthen anything. So it's not. This isn't just like neurotransmission. This is its. The nucleus has cells. These have to be the cells that the glial cells, perhaps. Also, is it the glial cells' job to carry these new proteins to the dendrite tips. Flow through the neuron. They only stick to the synapses that have the active tag. Or are these proteins literally flowing through the dendrite backwards from the normal direction of neurotransmission? All right, untagged synapses ignore the proteins. Tagged synapses capture them and use that. So when there's logic like this where it says like untagged synapses ignore the proteins and tagged synapses capture them, the tag was some chemical. So what I imagine is happening when these proteins get there is some chemical reaction that's actually affecting this growth. It's not like there's some extra logic now that's saying if tag then do this. It's that literally these systems just kind of play itself out in this chemical process to build permanent structure reinforcements, enlarging the spine, cementing the receptors. All right, the flashbulb effect, retrospective memory. Because the tag lasts for an hour, the timing can be reversed. You see something boring, synapse tag. 30 minutes later, something amazing happens. Dopamine release. The dopamine triggers protein synthesis. Those proteins travel back to the boring synapse and solidify that memory too. Whoa. Whoa, whoa, whoa. This is why you remember the trivial details leading up to a car crash. The crash, dopamine, solidifies the tags set by the boring drive minutes earlier. That's cool. So I think that strengthens my hypothesis here. Storm damage. Okay, let's let it ride. All right, so a dip or pause in baseline dopamine fire. This acts as a punishment signal, weakening the synaptic connections and discouraging the behavior. So why would this be the case? Also, if tagging seems to be 0 plus, I don't know. How do I say this? Or positive, then how does negative reward prediction error result in a punishment signal that ends up weakening synaptic connections? All right, then 0 reward prediction error, so everything went as expected, no change in firing rate. This implies that perceiving a predictable reward maintains behavior but does not spike dopamine. Implications for motivation. To maintain high dopaminergic output over time, a system requires intermittent reinforcement or the reframing of the effort itself as the reward. Relying solely on predictable external outcomes leads to the habituation of the DA response and a subsequent drop in motivation. Okay. All right, you have just derived a cutting-edge learning protocol known in neuroscience as behavioral tagging. Does this exist? Is this real? Oh, no. Okay, but this isn't that cutting edge. 2009 is a general mechanism of long-term memory formation. Memories are intertwined events. Little is known about the mechanisms involved in their interactions. Using 2 hippocampus-dependent and 1 hippocampus-independent learning tasks, we showed that in rats subjected to weak training protocols that induce solely short-term memory, long-term memory, protocols that induce solely short-term memory. Long-term memory is promoted and formed only if training sessions took place in contingents with a novel but not familiar experience occurring during a critical time window around training. This process, I imagine, has to be slightly after, right? This process requires new synthesized proteins induced by novelty and reveals a general mechanism of long-term memory formation that begins with the setting of a learning tag established by a weak training. These findings represent the first comprehensive set of evidences indicating the existence of a behavioral tagging process that, in analogy to the synaptic tagging and capture process, need the creation of a transient protein synthesis independent and input specific tag. All right, so that's effectively saying, I think, what we're getting at. But this, okay, a behavioral tagging account of kinase contribution to memory formation after spaced aversion training. 2 spaced weak inhibitory avoidance sessions induce long-term memory in rodents. I don't know what that means, though. Long-term memory can be induced by repeated spaced training trials. Oh, I see. Spaced training. I see. Space is like 2 spaced apart week sessions. So this is kind of that idea of doing something multiple times in a day, spaced out, but perhaps short, like perhaps week. I don't know what weak inhibitory avoidance though means. Using the weak inhibitory avoidance task, we showed that 1 session does not lead to a 24-hour long-term memory, whereas 2 identical sessions spaced by 15 minutes to 6 hours induce a 24-hour, induce the 24-hour long-term. This long-term memory promotion depends both on hippocampal protein synthesis and the activity of several kinases. In agreement with the behavioral tagging hypothesis, our results suggest that the 2 training sessions induce transient learning tags and lead via a cooperative effect to the synthesis of plasticity-related proteins that become available and captured by the tag from the second session. All right, that's pretty cool. What do we got? What do we got? Environment enrichment facilitates long-term memory consolidation through behavioral tagging. So what is environment enrichment here? Is another key experimental paradigm to explore the fundamentals of brain functioning. They've highlighted the importance of EE in enhancing cognition. Does it tell us what EE is? No. Novelty exposure is a decisive step in activating the molecular machinery of memory formation. Okay, so this has been like a topic. Yes, you can absolutely game the system. In fact, labs have demonstrated this in animals and increasingly in humans, where a weak memory studying is made permanent by a novel or rewarding experience, dopamine, that happens after the study session. Here is the breakdown of how to hack the tagging and capture system and how the proteins actually travel. Hacking the 60-minute window. Is it possible to exercise neurons and then immediately do something highly stimulating? Yes, this is effectively rescuing a weak memory. You study a complex topic for 30 minutes, the synapses are tagged, but the memory is fragile because you are bored. Within the 60-minute window, you can do something novel or unexpectedly rewarding, jump in a cold shower, explore a new environment, or win a quick game. The novelty triggers the VTA to flood the system with dopamine. The nucleus creates plasticity proteins. These proteins travel down the dendrites and are captured by the math tags you set 30 minutes ago. I was studying math. Did they say that? Okay. I think I just added that detail now. The math memory is consolidated, even though the math itself wasn't the exciting part. Do you need to think about it again? It helps, but strictly speaking, you do not have to. Oh my gosh, what is happening here? Should I just start gaming after these things? All right, the tag is physical. The tag is a chemical marker on the spine itself. It sits there waiting. However, briefly recalling the information, thinking about it, refreshes the tag, resetting the 60-minute timer. This maximizes the chance that the proteins will find it. The strategy, the optimal protocol is study, brief gap, novelty/dopamine. Warning on interference. The one risk is if the high dopamine activity uses the same exact neural circuits as the studying. If you study French and then immediately play a language-heavy video game, the game might overwrite the French tags. Physical novelty, going for a run in a new place, is often safer than cognitive novelty. Ah, okay. How did the proteins get there? The railroad. You asked, is it the glial cells or are they flowing through the dendrites? It is not the glial cells. The neuron handles its own logistics using an internal railroad system. The microtubule highway. Proteins are synthesized in the soma near the nucleus. They are then packaged into cargo vesicles and attached to motor proteins called kinesin. The direction. Kinesin walks physically along the microtubules, away from the nucleus and into the dendrites. Is it backwards? Not really. The signals flow. Have I been wrong about this? Has it always been this? Oh, no, wait. Signal flow, axon, synapse, dendrite, supply flow, soma, dendrite. Am I going crazy? Has it not always been dendrites being the input and then axons being the long range? All right, let's do it. Because dendrites, this is where logic would happen. Like there's dendritic logic occurring here. Received from other cells at synapses are transmitted to the cell body. Okay. So I gotta figure out what's going on here. Signal flow. My understanding was that normal neurotransmission goes synapse to dendrites where logic can occur based on other synapse inputs to cell body to axon to synapse. Significantly longer dendrites. Is it backwards? Not really. So the proteins are flowing towards the input site to reinforce it. Right, okay, towards the input site. When I say that the protein railroad direction is backwards, I'm just pointing out that it's moving in the opposite direction as neurotransmission signals. All right. The modern twist. Local synthesis, the 3D printer. Biology found that that shipping proteins from the nucleus is too slow. It takes hours. The solution, the neuron doesn't just ship finished proteins. It ships the blueprints, mRNA, and the factories, ribosomes, out to the dendrites before they are needed. Okay, this is stuff I recall. Parked mode. These factories sit dormant at the base of the dendritic spine. Activation. When dopamine and glutamate hit the synapse, they turn on local factory. The structural proteins are built on... Okay, the next question. Oh, man. I jumped the gun. Also, does the... does a neuron send proteins or RNA, mRNA slash, what was it, ribosomes? Is that what we're talking? Ribosomes to the axon as well? Or is that usually too far away? When dopamine and glutamate hit this... Okay, parked mode. These factories sit dormant at the base of the dendritic spine. When dopamine and glutamate hit the synapse, they turn on the local factory. The structural proteins are built on site in minutes rather than waiting for delivery from the nucleus. The gamer's protocol. If you want to use this biology to learn faster, set the tags. You focus intensely for 20 to 40 minutes. Trigger the global signal within 1 hour. Induce a phasic dopamine spike. Novelty, cold exposure, or an unexpected reward. Avoid competition. Do not engage in a task that requires the exact same processing loop immediately. Let the proteins find the tags you just set. Okay, another question then. What leads to tag overwriting? Actually, this is kind of a similar question to this last one. Similarly, what leads to tag overwriting? How would the use of similar neurons result in previous tagging to go away? All right, this is crazy stuff. All right, D1 versus D2 receptors, stability versus flexibility in the prefrontal cortex. In the prefrontal cortex, the effect of dopamine on cognitive focus depends on which receptor subtype is activated. This creates a stability-flexibility trade-off. So D1 receptors, stability, these receptors are located on the postsynaptic membrane of excitatory pyramidal neurons. D1 activation enhances the NMDA and AMPA receptor currents, effectively locking in the sustained firing required for working memory. High levels of D1 signaling are associated with robust focus and resistance to distraction. Now, D2 receptors, for flexibility, these are often located on inhibitory interneurons or presynaptically autoreceptors. D2 activation tends to suppress activity or destabilize the current network state. This is crucial for cognitive flexibility, the ability to switch tasks or update rules. However, excessive D2 dominance or insufficient D1 leads to a fluid mind that cannot hold a thought, a hallmark of ADHD or mania. All right, mechanism of SNR enhancement, signal-to-noise ratio enhancement. Dopamine in the prefrontal cortex enhances the signal-to-noise ratio through a dual mechanism, boosting signal and suppressing noise. Okay, that makes sense. So, yeah, I'm wondering if this is the mechanism to answer the question I just asked. Okay, the backwards railroad, you're absolutely correct. Information flows, dendrite, soma, axon. That's electrical, but this is a supply chain flow, of a physical supply chain flow where it goes soma to dendrite and soma to axon. All right. So yes, the construction crews, proteins and kinesin. Let's actually figure this one out. Pronounce. Kinesin are effectively driving upstream against the flow of electrical traffic to reach the dendritic synapses. Okay. Do they ship to the axon, the long haul? Yes, absolutely. For a long time, neuroscientists thought the axon was just a dumb wire that couldn't build anything. Now, we now know that was wrong. The neuron ships both finished proteins and mRNA blueprints all the way down to the axon terminals. Why it's harder? Axons can be incredibly long. For example, the nerve running from your spine to your big toe is 1 meter long. Shipping a protein there from the nucleus would take days. Wow. So the axon terminal has its own local 3D printers, the ribosomes, and a stockpile of mRNA, the instructions. This is critical for presynaptic plasticity. The axon needs to change how much neurotransmitter it releases, for example, building more vesicles or calcium channels. It can't wait for the CEO, the nucleus, to send supplies, so it builds them on site. All right, how does negative reward, the dip, weaken a connection? This is one of the most elegant mechanisms in the brain. You asked if it's just a lack of signal or a specific punishment signal. It is a specific inverted signal. When you expect a reward and don't get it, negative reward prediction error. Dopamine neurons don't just stop firing, they pause their background tonic firing. This dip triggers a specific chemical chain reaction that actively dismantling the synapse. Is this just a typo on Gemini? That actively, or maybe it's from the quote here. Actively dismantles the synapse. The mechanism kinases versus phosphatases. I don't know. Phosphatases. Think of the synapse as a wall being painted. We got the painters. They add phosphate groups to proteins. This makes the synapse stronger. And then we got the strippers. Ayo, they remove phosphate groups. This makes the synapse weaker. So tug of war, high dopamine reward, activates protein kinase A. PKA screams, paint the wall, and also handcuffs the strippers. Jeez, what has happened here? Inhibits the painters. I'm sorry, the strippers here. And we got strong synapse. The dip, disappointment. When dopamine drops to 0, the PKA shuts off. The handcuffs come off the phosphatases. The result, the strippers, specifically calcineurin, immediately attack the synapse. They physically rip the AMPA receptor off the surface and drag them inside the cell. The connection is not just less strengthened, it is actively weakened. All right. Oh, man. Okay, I think the takeaway here is simply that a drop in dopamine is going to actually change how much dopamine is being processed through these neurons, which is going to affect a chain reaction, a chemical imbalance that results to damage or results in damage. But extra dopamine is going to result in increased building. So I think that's what's happening here. All right, how does overwriting work? You asked how a new activity could overwrite a previous tag. This happens through synaptic competition. Imagine the neuron has a limited budget of construction supplies, so plasticity-related proteins floating around. In scenario A, consolidation, you study math, you set tags, you rest, the neuron makes supplies, the math tags grab the supplies. Interference, you study math, immediately afterwards you play a high-intensity video game, and that's setting new tags. Because the video game was more stimulating, the neuron prints supplies, but the video game tags are stickier. Stronger electrochemical affinity. So, okay, does that competition pretty much nullify my hypothesis about gaming the system? Why would it matter if the subject matter was significantly different? Wouldn't that different subject matter still create stronger tags? Okay, so it's a competition. That's one thing that we're seeing. All right, the next thing, the inhibitory breaks, serotonin and satiety. I don't know, how do we say this? I know satiate, but
satiety.
Satiety. All right. Satiety. If dopamine is the accelerator, serotonin is the brake, originating from the RAF nuclei in the brainstem. Serotonergic projections permeate the brain, regulating mood, patience, and satiety. The interaction between 5-HT and DA dopamine is... Oh, this is serotonin. All right, so the interaction between serotonin and dopamine is predominantly antagonistic, forming a yin-yang relationship in the regulation of motivation. Okay. All right, it does not nullify your hypothesis. In fact, it explains exactly why your hypothesis works, provided you understand one critical biological rule. Competition for proteins only happens inside a single neuron. Oh. Whoa. So when it says, so when you say proteins are floating around, you don't mean in the space in between neurons, such that any synapse could pick this up within that space. You mean within the dendrite. Such that any dendrite end / synapse could pick it up. Is that correct? Okay. If math uses neuron, okay, so this is all that. It's the same neuron rule. Are synthesized inside the cell body and float down the dendrites of that specific cell. They cannot jump from neuron A. Okay. Why pure novelty is best. This is why the best hack is an activity that provides high dopamine but low cognitive load. Cold shower, sprinting, huge dopamine release, but very little complex information to encode. Complex video games, high dopamine, but also high tagging. Learning maps, strategies, etc. Even if the circuits are different, you are taxing the brain's overall metabolic energy. But more importantly, if the game requires focus or working memory, you might start overlapping with the neurons you just used to study. Summary, how to game the system. You are not nullified. You just have to follow the rules of territory. Visualizing the global bath. Think of dopamine like the AC turning on in a massive office building. The dopamine cools the entire building. The competition, if everyone is crowded into conference room A, they fight over who gets the standard. Okay. If half the people are in A and half are in B, everyone gets cool air comfortably. Nice analogy. Nice metaphor. All right, when I say floating around, I mean intracellularly, inside the neuron's own skin. I do not mean extracellularly, in the gap between neurons. Okay. Let's move on. In the nucleus accumbens in VTA, serotonin acts to restrain dopaminergic drive. This is the neurochemical basis of satiety and quiescence. And quiescence means inactivity or dormancy. Okay. Serotonin released in the NACC binds to, serotonin receptors. These receptors are located on the GABAergic interneurons or directly on DA terminals. So this is what, inhibitory? GABA. Activation of these receptors inhibits the release of dopamine. Nice. Behavioral outcome. This inhibition signals enough. It is the transition from wanting to having. When serotonin levels are low, the brake is removed, leading to impulsivity, aggression, and an inability to wait for delayed rewards. Sexual satiety. This mechanism is clearly demonstrated in the I'm not going to say that word, interval. Following sexual climax, a massive release of serotonin in the lateral hypothalamus and NACC inhibits DA release, inducing a state of quiescence and non-responsiveness to further stimuli. Wow. All right. Satiety signaling. The gut-brain axis. Serotonin is also the primary messenger of metabolic satiety. Peripheral to central. Feeding triggers the release of serotonin in the gut and brainstem. Feeding. This serotonin activates pro-opiomelanocortin neurons in the arcuate nucleus of the hypothalamus. So, POMC versus dopamine. POMC neurons project to the reward centers and inhibit the dopaminergic response to food cues. This creates a negative feedback loop. As calories are consumed, serotonin and POMC levels rise, dampening the reward value of further food. That's cool. In obesity or addiction, the serotonergic brake may fail or the dopamine system may become hypersensitized, leading to a superstimulus effect where satiety signals cannot overcome the drive to consume. Impulsivity in the weighting system. Serotonin is crucial for active weighting or impulse control. So dopamine depletion leads to impulsive action. Serotonin depletion leads to impulsive action and an inability to tolerate delay. Animals with low serotonin will choose a small immediate reward over a large delayed reward. Serotonin innervation of the what is innervation again? Innervate, that's supply of nerves to body part. Versus enervation? Is this not a thing? Enervation means a serious weakening or loss. Okay, never mind. Serotonin innervation of the orbitofrontal cortex and NACC helps to encode the value of the delay, effectively lowering the discount rate of future rewards. It allows the organism to endure the passage of time without abandoning the goal. Okay. The value of the delay. That's pretty neat. So that's kind of a meditation thing as well. You could think of it as you're just in a delay. A delay of the automatic barrage. And maybe this can help you, high serotonin helps you value that. All right, the metabolic substrate. Glucose, lactate, and the astrocyte. The user query specifically asks about the deployment of glucose. The brain is a voracious consumer of energy, accounting for about 20% of the body's glucose use, despite being only 2% of its mass. However, the model of neurons simply passively absorbing glucose from the blood is outdated. Current research points to a dynamic partnership between neurons and glial cells, astrocytes, known as the astrocyte neuron lactate shuttle. The astrocyte neuron lactate shuttle. When the brain enters a state of high focus or intense computation, for example, maintaining gamma oscillations, the energy demand of neurons exceeds the rate at which they can metabolize glucose via oxidative phosphorylation alone. Glutamate trigger. Active neurons release glutamate. This glutamate is taken up by astrocytes via transporters. The uptake of glutamate triggers the astrocyte to absorb glucose from the bloodstream. Crucially, the astrocyte does not burn this glucose completely. It processes it via aerobic glycolysis into lactate. The lactate is then transported out of the astrocyte and into the neuron. Inside the neuron, lactate is converted to pyruvate, which enters the mitochondria to generate ATP rapidly. So what has happened here? A chain reaction has happened that leads to the neuron getting more energy. But what just happened here? Okay, glutamate. So the neurons are sending a signal out saying, hey, we're going to need more energy. We're active. We need more energy. And once again, this isn't like some separate logic. It's literally like active neurons send out this signal. And yeah, these cells that are close by start absorbing glucose from the bloodstream. So, okay. I'm learning about the astrocyte neuron lactate shuttle system. What is the distribution of blood within the brain? How are astrocytes able to readily get glucose from the blood? From blood, right? This is what's going on from the bloodstream. I'll just say that. And then, yeah, this is all just stuff that, and then, okay, and it's not that the neuron's actually getting glucose. It's the astrocyte is preparing. It's translating that energy into something that the neuron can use. So what? Okay, the model of neurons simply passively absorbing glucose from the blood is outdated. I see. To understand why the ANLS works, you have to realize that neurons are actually quite lazy when it comes to sourcing their own food. They delegate the logistics to astrocytes. The distribution of blood in the brain is not like a river. It is like a dense, pervasive mesh. The brain is packed with capillaries in the cortex, gray matter. The density is so high that no neuron is more than 20 microns away from a blood vessel. For context, a neuron cell body is about 10 to 20 microns wide. So the surface area, if you flatten out all the capillaries in your brain, you would cover a tennis court. Crazy. We used to think of neurons and blood vessels as separate. We now treat them as a single functional unit. The blood vessel doesn't just pass by, it is physically tethered to the neurons and glia. How astrocytes get first dibs, the physical monopoly, is that they physically strangle the blood vessels. Whoa. If you look at brain capillary under a microscope, you won't see the blood vessel wall directly. It is completely covered, almost 99% surface area covered by astrocyte end feet. What? This creates a second barrier. For glucose to leave the blood and enter the brain, it often has to pass through or between these astrocyte guards. Astrocytes are the gatekeepers. They are physically positioned first in line. The glucose hits the astrocyte end foot before it ever sees a neuron. Nice. All right, when people talk about the blood-brain barrier, what are they referring to? The readiness of the glucose uptake is also chemical. The astrocyte uses a specific transporter that matches the blood vessel's transporter. Okay, yeah, they're using the right chemicals to pull the glucose out. And then it's preparing it for the neurons. Oh, wow, this is, okay, high capacity designed for bulk transport, but this is a high affinity. That's a good reason why. The active control, vasodilation. Astrocytes don't just wait for glucose, they demand it. When a neuron fires, the astrocyte detects it. The astrocyte then releases vasodilators directly onto the blood vessels it is holding. The blood vessels widen. The blood vessel widens, dilation, bringing more glucose specifically to that astrocyte's location. That's cool. That's cool. All right, when people talk about the blood-brain barrier, they're referring to a rigorous security checkpoint that separates your circulating blood from the sensitive environment of your brain tissue. Physically, it's not a separate shield or membrane wrapped around the brain. Instead, the barrier is the blood vessel itself. Here is exactly what makes brain capillaries different from capillaries in the rest of your body. The bricks, specialized endothelial cells in the rest of your body, like your muscles or liver. The cells that form the walls of blood vessels are somewhat loose. They have small gaps that allow water, nutrients, and small molecules to leak out freely. In the brain, these cells are radically different. No gaps. They are fused together tightly. They rarely drink or swallow fluid from the blood to move it to the other side. Okay, low pinocytosis. I think I need to know that concept in order to understand what this is saying. Efflux pumps, they have built-in ejector seats that actively grab toxins, trying to cross and throw them back into the bloodstream. Oh, that's cool. All right, the mortar, tight junctions. This is the most critical component. The cells are stitched together by protein complexes called tight junctions. Think of these as a waterproof zipper. They seal space between the cells so effectively that not even ions or water molecules can slip through the cracks. To get into the brain, a molecule cannot go between the cells. It must go through them. This forces everything to use a specific key transporter. So, yeah, okay, we need this VIP list. Only certain things can get through. These cells provide the wall, but they don't work alone. They are maintained by the guards, as we discussed earlier. These cells sit embedded in the basement membrane of the capillary. They regulate blood flow and signal the wall cells to keep the tight junction zipped up. If they die, the barrier becomes leaky, as in diabetic retinopathy. As you notice, these wrap the vessel. They release chemical signals that tell the endothelial cells, these are the wall cells, you are in the brain now, build the wall higher. All right, what actually gets through? The VIP list. Because the zipper is closed, there are only two ways to cross. The ghost method, lipid solubility. If a molecule is tiny and fat soluble, it can simply diffuse right through the cell wall like a ghost. Okay. So this is like oxygen, CO2, alcohol, nicotine, caffeine. These are, I'm guessing these are why these things can get into our system really quickly. This is why these drugs affect you so quickly. The key card method, specific transporters. If a molecule is large or water soluble, polar, it needs a dedicated transport protein to carry it. Glucose, we got the GLUT1 transporter as discussed. Amino acids, we got a transporter. The problem for pharma, 98% of potential drugs cannot cross the blood-brain barrier because they are not lipid soluble and don't have a dedicated transporter. This is the biggest hurdle in treating brain diseases like Alzheimer's. Okay. Good to know. Can we get a nasty look of like capillaries in the brain? Like, can we see that? Oh, look at that. It needs to be much more dense, right? Like it needs to be pervasive. Am I right or am I wrong about this? Or do neurons transport any of this stuff? Okay, this is what we just learned about. All right, let's continue here. All right, lactate acts as a turbo fuel. It can be metabolized faster. So this is what, GLUT3? Is that lactate? Is lactate GLUT3? Is that GLUT3? I don't know. The fuel and byproduct. I see. And this is the glucose grabber. So, okay, GLUT3 is the transport here. Okay, okay. That's, yeah, these are talking about transporter. This is the actual product. All right. Glycogen, the brain's reserve tank. Unlike most of the body where glycogen is stored in muscles/liver, in the brain, glycogen is stored almost exclusively in astrocytes. Okay. The release of norepinephrine during arousal and vasoactive intestinal peptide stimulates the breakdown of astrocytic glycogen into lactate. Okay, so they've got reserves. Oh my gosh, yep, this is the reserve tank. This provides an immediate energy buffer for the neurons before blood flow can increase to meet demands. Nice. I'm curious how they figure this stuff out. Like, are they literally, do they have to find an astrocyte, put it on a petri dish? Do they have to just look, or do they have to put it into some machine that tells them what kind of molecules are in there? Like, what's the how do you figure this stuff out? Memory formations. This mobilization of glycogen lactate is essential for long-term potentiation. Blocking glycogenolysis in astrocytes prevents the formation of long-term memories, proving that learning is an energy-dependent process constrained by metabolic availability. All right. Cortisol, the metabolic mobilizer, the primary stress hormone, plays a critical role in ensuring this fuel supply is available. Acute stress, the good. In the short term, cortisol creates a permissive environment for high performance. It inhibits insulin-mediated glucose uptake in peripheral tissues, like muscles and fat, effectively saving the glucose for the brain. It also stimulates gluconeogenesis in the liver, increasing blood sugar levels. This ensures that the high demand has sufficient substrate. Chronic stress, however, prolonged elevation of cortisol becomes neurotoxic. Chronic glucocorticoid exposure causes the atrophy of dendritic spines in the prefrontal cortex and hippocampus, reducing the brain's capacity for focus and memory. It essentially burns out the machinery it is meant to fuel. Okay, that seems fine. That checks out. Adenosine, the metabolic cost of thinking. All right, every molecule of ATP used for focus creates a byproduct, adenosine. Accumulation as ATP is hydrolyzed to ADP, AMP, and finally adenosine. This molecule accumulates in the extracellular space. Oh, okay. Adenosine is the thing that makes it so that it's hard for us to focus, right? The fatigue signal. Adenosine acts as a homeostatic fatigue monitor. It binds to A1 receptors on presynaptic terminals, inhibiting the release of excitatory neurotransmitters, glutamate, acetylcholine, and dopamine. It effectively dims the system to force recovery. Sleep pressure. This accumulation creates sleep pressure. Sleep, and specifically non-REM deep sleep. is the only process that efficiently clears adenosine, restoring the brain's capacity for ATP generation and high frequency firing. So this is where sleep is so important, or else you're not getting any of that back. Caffeine works by antagonizing, blocking adenosine receptors. It does not give energy. It merely blocks the fatigue signal, allowing the catecholamine circuits to continue. Oh wait, the circuit DA and norepinephrine, dopamine, norepinephrine, to continue driving the system despite the metabolic debt. Caffeine works by antagonizing, blocking adenosine receptors. Whoa. It does not give energy, it merely blocks the fatigue signal. Wow. But then it also, obviously, depending when you drink it, can affect sleep in such a way where you don't clear those things out. All right, tuning the signal, GABA, glutamate, and EI balance. While modulators like dopamine and acetylcholine set the tone, the actual information processing in the brain is carried out by the fast-acting neurotransmitters glutamate and GABA, right? The precise balance between these two is what determines the resolution of attention. So we got parvalbumin, interneurons, and gamma oscillations. Attention is not a static beam, it is a rhythmic pulsation, specifically in the gamma band. This rhythm is generated by whatever the heck that is, interneurons. How do these differ from the claustrum in also in a functional sense? All right, these interneurons are fast spiking cells that provide strong, precise inhibition to the soma of pyramidal neurons. By firing rhythmically, they create windows of excitability. Pyramidal neurons can only fire when the PV plus inhibition momentarily lifts. This synchronizes the firing of thousands of pyramidal cells, creating a coherent chorus that can be heard by downstream regions. Focus mechanism. When we focus, PV plus neurons work harder, tightening the synchronization. This increases the efficiency of information transfer. All right, this is a really important part of the system. Dysfunction. In conditions like schizophrenia and autism, PV plus interneuron function is often impaired. This leads to gamma asynchronous, a state where the brain is noisy and unable to bind features into a coherent whole or filter out distractions. All right. This is a nice comparison to make. Both PV plus interneurons and claustrum are essential for controlling the flow of information in the brain, and both are heavily implicated in consciousness. However, they operate on completely different scales and use different strategies. Simplest way to think of it is PV plus interneurons are the metronomes, local timing. The claustrum is the conductor, the global coordinator. Here's the detailed breakdown and how they differ. The scale, local, okay. The local police, they are scattered diffusely throughout the entire cortex. They are embedded agents. They have short axons. They only affect their immediate neighbors. They manage the activity of a single cortical column. They ensure that when column A fires, it fires cleanly and precisely. The claustrum is the central switchboard. It's a thin sheet of gray matter tucked deep in the brain between the insula and putamen. It has massive long-range projections that reach almost every part of the cortex simultaneously. It coordinates activity between regions. It decides when the visual cortex should talk to the parietal cortex. Okay. So this is coordination, but it's not actually affecting the frequency. The mechanism, direct versus indirect inhibition. The claustrum actually sends glutamate signals to the cortex. These excitatory signals specifically target local interneurons in the cortex. Oh, often PV plus cells. Okay, there we go. So these excitatory signals could actually excite the inhibitory neurons, which are going to set the tempo. So these guys, yeah, this is the boss here. Colostrum excites the local police to shut down the noise. It uses the local PV plus cells as its enforcers. This allows the colostrum to silence broad areas of the brain so that one specific area can stand out. Selective attention. You mix that with more localized mechanisms of acetylcholine, and you've got yourself a hypersensitive, focused, conscious experience. All right, gamma versus slow wave. Both are involved in oscillations, but they control different frequencies. PV plus interneurons, high frequency. Colostrum, the slow wave coordinator. Slower frequency. The colostrum seems to coordinate the down states across the entire brain. This is crucial for sleep, but also for task switching. Okay. Okay, so failure state, schizophrenia, if PV plus cells fail, gamma becomes muddy, you cannot distinguish signal from noise. But if you lose the colostrum, you lose consciousness. If you electrically stimulate the colostrum in epilepsy patients, consciousness instantly shuts off, the system crashes. Yikes. Major yikes. Are we going to talk about that here, or what? What's going on here? Okay, glutamate accumulation and cognitive fatigue. Why does thinking hard feel exhausting? Recent research points to glutamate accumulation as a key driver of cognitive fatigue. So I could have sworn that it would have been the, was it adenosine? What was the other thing that was building up? Did we not just see this? The fatigue signal. Adenosine. Yo, the director, what's good? Welcome back. We're doing some like real deep in the weeds stuff. I have to learn all these molecules that people always use when talking about the brain and focus and stuff. These are all the Andrew Huberman players, the usuals, the usual suspects. Okay, glutamate accumulation and cognitive fatigue. Recent research points to glutamate accumulation as a key driver of cognitive fatigue. Glutamate is excitatory, but in excess it is toxic. It must be tightly regulated and cleared by astrocytes. Oh. Okay, so astrocytes are also clearing glutamate. Right. Okay, glutamate needs to be cleared. So the astrocytes take the glutamate, they go to the blood, they get the other stuff, they bring it back, you got energy. But if there's too much, then the astrocytes are unable to complete their orders, and you've got a problem. During prolonged high-demand cognitive tasks, for example, 6+ hours of intense focus, glutamate accumulates in the extrasynaptic space of the lateral prefrontal cortex. This accumulation makes the lateral prefrontal cortex harder to activate. To protect itself from excitotoxicity, the brain essentially throttles down the lateral prefrontal cortex. Since the lateral prefrontal cortex is responsible for impulse control and long-term planning, its fatigue leads to a shift in behavior. The tired brain defaults to low-effort, immediate reward choices. Impulsivity. Quite biological, isn't it? It is quite biological. Yeah, all this stuff, it comes down to like a chemical experiment, just like chemical processes. It's literally just a it's just things happening, It's just these things rubbing up against each other, transforming into something else, and that causes some chain reaction. It's kind of wild. And the crazy thing is, like, all the logic that happens in our brain is just literally what it's like a pragmatic view. It's just what happens. It's, you know, all these chemical reactions happen in such a complex way that you get logic. All right, so what am I asking here? I'm saying if glutamate, what's the difference between cognitive fatigue from glutamate toxicity and what was the other one we said? And fatigue from adenosine. Or is adenosine what's leveraged when there is glutamate toxicity to tell neurons to stop firing? I'm currently focusing on cybernetics, especially Gregory Bateson. I do not know him. Actually, I need to know cybernetics a little more. Cybernetics, what do we got here? Transdisciplinary science of control and communication in complex systems. Nice. Focusing on circular, self-regulating processes like feedback loops found in animals, machines, and society to achieve goals. Whoa. Very cool. What's been the focus of interest? Like, what's the is there a sub like a sub area that you're focused on, or is this all in pursuit of some larger focus that you require cybernetics to better understand this larger picture, or to find some solution to, or some mechanism for the larger picture? All right, you're essentially asking the difference between running out of gas, adenosine, and the engine overheating, glutamate. I see. While they happen at the same time, they are biologically distinct signals, and your intuition is correct. Adenosine is the emergency brake the brain pulls to prevent glutamate toxicity. Adenosine fatigue, drowsiness, heavy eyelids, yawning, the physical urge to lose consciousness. Metabolic debt, you burned too much ATP. Global, it diffuses everywhere, affecting overall arousal. And sleep, you need to rebuild ATP. Glutamate fatigue, I can't think. You aren't sleepy, but you feel resistant to doing one more math problem. It is executive failure. Synaptic, okay, so when we talked about adenosine, we talked about sleep, right? Where you need to get this stuff back. I'm going way too far back now. Is the only process that officially clears adenosine. Okay, so adenosine. So an overload of adenosine cannot be rectified except for deep sleep, whereas glutamate toxicity can be alleviated over time, I imagine, as what are those things called? Acetyl. We literally just talked about it. Ba 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 ba. What are those glial cells called? I don't remember. Yo, Energy Dominator, what's good? I'm a filmmaker / student, so at the end, I always relate to media theory, the role of the speculator, etc. Oh, that's really cool. Like at the end of your project, are you saying? Or are you saying at the end of the day, like that's something you generally do? What is good, Energy Dompter? What's been popping? Okay, we got was it is it at Astrocyte? Yep, okay. Astrocytes. Spotlight of Attention 3, we're on 4, right? Yeah, the breaks. Nope, we got to this as well. We got to section 5. Okay. As the astrocytes continue using the glutamate to get energy from the blood, so can... Oh, no. Okay. I wrote that probably. I mean, I always try to relate the theory from other fields to mine. Okay, I see. Yeah, I mean, that's the way to go. That's like the learning tip, or the learning secret to success, the special sauce, to just try to have something to relate the new information to. Makes it so much more easy to remember and salient. I've recently been thinking about fragmentalism and how that relates to Einstein's theory of special relativity. Are you familiar with the concept of fragmentalism? Out of curiosity, I'm not. Fragmentalism is a philosophical view often in metaphysics and philosophy of time, proposing that reality isn't a single unified whole, but is composed of distinct, internally coherent fragments of facts that are incompatible with each other across fragments, like different moments in time or different perspectives, without a single overarching viewpoint. Well, I guess our view is kind of a fragmentalism. Like, I kind of point out that the way that we can think about the semantic facts that we understand is that they would be changing from moment to moment as as these facts are manifesting because of the brain connections that are constantly being strengthened and diminished. So like the shape of the structure of our brain is constantly changing, and that leads me to believe that the way that we think about things, even facts, would differ over time. It would change. Everything is anything that you think about is being changed by the process of you thinking about it. So there's nowhere you can run in some sense where you're not dealing with a thought that isn't changing connections. So yeah, you could think about it that way. I mean, this is just a metaphysical way we can conceptualize information that we know over time. But the thing that the thing I want to point out is that it wouldn't be appropriate to like take this view and to then like blow it up to multiple people. Like, each of us have a different understanding that is constantly changing over time. So the semantic facts that we each understand or we think are true are changing over time, but they're different from, like, each of us has a different understanding. So the fragmentalism view, in my mind, is something that you can only use for a single person. Like, it wouldn't make sense to use it across people. Now, that being said, I don't know how we would, like, the way that I just said that, I don't know how we would connect it to Einstein's theory of special relativity, because these are just different physical systems that we're talking about at that point. I'm talking about the brain physical system, and the brain doesn't really care about special relativity. But yeah, the fragmentalism, like how as it's described here, is ambiguous enough that you could think about it in many different ways. The way I just described is just the most intuitive that I can think of, that makes sense of the like what we know about the brain and how that affects our experience. and understanding. All right, you've hit the nail on the head regarding the difference in remediation. Adenosine overload requires a state change. It requires sleep. You generally cannot rest your way out of adenosine pressure while awake. So this is why you can't just like get awake again unless you drink caffeine, which inhibits the adenosine receptors. Glutamate buildup requires, or you can just straight up overload the system with like amphetamines. All right, glutamate buildup requires a activity change, time, low focus. You can clear this. You can clear this while awake simply by stopping the specific task that caused it. Okay. Why you can't clear it while awake? Adenosine accumulates in the extracellular space. Oh, when you are awake, your brain cells are physically swollen with fluid to support their high activity. They take up so much space that the sewage pipes between them are squeezed out. This is the brain's plumbing. It flushes cerebrospinal fluid through the brain to wash away waste, like adenosine. Okay, I see. When you enter deep sleep, your brain cells effectively shrink by 60%. Wow. The shrinkage opens the floodgates. CSF, cerebrospinal fluid, rushes through the widened gaps, physically washing the adenosine away. While you are awake, even if you are resting on the couch, your cells remain swollen and the plumbing remains closed. You cannot flush the system until you lose consciousness. Wow. And then glutamate, it just requires someone to clean up. You are correct that this can be alleviated over time without sleep, but there is a slight correction on how the astrocytes do it. Cleaning glutamate costs energy. You imagine astrocytes using glutamate to get energy from the blood is actually the opposite. Cleaning up glutamate is an energy expensive chore. Astrocytes are massive amounts of ATP to use massive amounts of ATP to power the pumps that suck glutamate out of the synapse. The recycling, they don't usually burn the glutamate for fuel. They convert it into a safe inert molecule called glutamine. That's a troll to call it that. Then they handle this glutamine back to the neuron to be used later, reused later. Why resting works for glutamate. Since this is a local problem, you just need to stop using that specific circuit. If you are studying, you are dumping glutamate faster than the janitor can sweep. The pile grows. Okay. Okay, so astrocytes will clean the glutamate if it's too much, and it will otherwise still carry out the process of using glutamate to get glucose from the blood, from what did we just call those things? I forget. Capillaries? I think that was the word. All right, unlike most of the body where glycogen is stored in muscles, liver, in the brain, glycogen is stored almost exclusively in astrocytes. This was the reserve tank. Cortisol, the metabolic mobilizer. Right. And then adenosine, the metabolic cost of thinking. I see. Okay. This accumulates. Okay, and then we got to tuning the signal. This is something we did. The metronome. Yep. Glutamate accumulation and cognitive fatigue. Right. It's during prolonged high demand cognitive tasks. Glutamate accumulates in the extrasynaptic space of the lateral prefrontal cortex. Blah, blah, blah. Okay. So we're at 7 now. Section 7, social and salience modulation, which I think we're going to chill out on for now. Oh my gosh, I've been going for an hour and a half. What the heck? I thought we just started. Oh, we got an archive. All right. That paper, to my understanding, discusses the relationship between special relativity, quantum physics, and fragmentation, fragmentalism. Right. I guess the idea of fragmentism that would be covered here, I would imagine, is like a much more metaphysical, like a broadly metaphysical theory about some objective truth in the universe, which I usually try to assault. But let's see what's going on here. Or I try to push back on, I should say. I should be less violent with my rhetoric about that. This paper examines how presentist fragmentalism. So what is present? Is a metaphysical and physical theory suggesting reality isn't a single continuous timeline, but a collection of discrete interconnected fragments, each with its own real unfolding now. Whoa. So this is really tricky, because like, what do we mean by a timeline or reality when we're talking about fragments? Like, this makes sense if you're saying that there's multiple people out there and each person is experiencing time in a different way. But this is going to connect it to like the universe's time. So this is actually kind of looking at some sort of, I don't know, panpsychism, if I'm understanding this correctly. Provides a unified framework for resolving key paradoxes in quantum mechanics and relativity. PF posits that reality consists of fragments with independent A-series temporal flows, dynamic future, past, present, connected by B-series relations. So unless this is actually like a straight up scientific theory about quantum physics and time, basically pointing out, let's just say, we're talking about a many worlds situation in which each of these worlds has a different sense of time. Okay, how does this type of fragmentalism contrast or perhaps resonate with, I can put the with outside, with the many worlds hypothesis? So that type of question is actually going to make it much easier for me to understand presentist fragmentalism, I think. We demonstrate PF's explanatory power through analysis of 8 fundamental thought experiments, including Einstein's train, Schrödinger's cat, EPR, and the delayed choice quantum eraser. The framework naturally resolves apparent contradictions between quantum non-locality and relativistic causality by distinguishing between B-series relations, which respect relativistic constraints, and A-series synchronization, which enables quantum correlations. This reconciliation requires no additional mathematical machinery beyond standard quantum mechanics, while providing clear ontological understanding of quantum phenomena. We compare presentist fragmentalism with other major interpretations, including Aharonov's two-time, Ravelli's relational interpretation and quantum reference frames, showing how PF offers unique insights into temporal structure. The framework's success in systematically resolving diverse paradoxes while preserving both relativistic and quantum principles suggests it captures fundamental features of physical reality. Based on the provided text, the author does not explicitly discuss the many worlds interpretation. The text focuses... Yeah, I understand. However, based on the specific definitions of presentist fragmentalism provided in the document, we can infer distinct contrasts and resonances with the standard many worlds hypothesis. All right, contrasts. The nature of outcomes in time. The most significant difference lies in how each theory handles measurement, outcomes, and the structure of time. So in PF, the text describes measurement as a process that produces determinate properties within a shared present moment. When fragments merge, they establish a single shared reality relative to one another. The evolution of one fragment relative to another is described as stochastic, probabilistic, because there is no fact of the matter prior to the merger. Aha. In contrast, in many worlds, all possible outcomes of a quantum measurement are realized in separate non-communicating branches of the universal wave function. There is typically no stochastic selection of a single outcome. All outcomes exist deterministically in the multiverse. Okay. I'm not going to be able to understand the differences here, but synchronization versus branching. PF measurement is defined as temporal synchronization where disparate A-series flows merge into one. This is a unification process. And in many worlds, measurement is typically viewed as a branching or decoherence process where a single history splits into many. Okay. Dynamic versus static time. PF, this theory relies heavily on the A-series of time, a dynamic flow from future through present to past. It posits that future possibilities actualize into present experiences. Okay, and many worlds is generally associated with a block universe, B-series only, or eternalism, where all times are equally real and there is no objective dynamic flow or privilege now. Oh my gosh, PF is working with Tenet rules on the A series of time. Does PF give any credence to the Tenet universe? I gotta do that just for fun. Okay, resonances, realism, and no collapse. Despite their differences, PF shares some philosophical DNA with many worlds regarding how they treat the wave function and the observer. Rejection of the standard collapse. The text states the theory requires no additional mathematical machinery beyond standard quantum mechanics. It resolves the collapse problem not by a physical collapse of the wave function, but by the synchronization of time, A-series, between previous unsynchronized fragments. Similarly, many worlds claims to use only standard quantum mechanics without adding a collapse postulate. Both theories attempt to explain the appearance of collapse without a traditional collapse mechanism. And then fragmented perspectives. Reality is composed of fragments where for one fragment there is no fact of the matter about the A-series values of another fragment. A system exists in a superposition relative to an observer because they do not yet share a now. And many worlds. This resonates with the many worlds view that the state of a system is relative to the observer's branch. In both views, there is no single absolute view from nowhere that captures the classical state of the entire universe at once. Reality is perspectival. Okay. Okay, PF does not give credence to the mechanics of the Tenet universe. In fact, the text explicitly rejects the core mechanism required for Tenet-style time inversion. Okay. Explicit rejection of retrocausality. The central premise of Tenet is that objects or information can be inverted to flow backwards through time, allowing the future to affect the past. The PF stance. The document states clearly regarding the delayed choice quantum eraser. The key insight is that there is no backwards causation. Instead of future measurements influencing past events, PF argues that the system remains in an indeterminate state relative to the observer until the A-series, time flows, of the fragments merge. The direction of temporal flow. Okay. All right, back to the actual stuff. What would each theory suggest about each person's conscious experience, and do either of the theories hold an not an axiom, make a conclusion, hopefully evidenced, or logical, that contradicts the other. Because so like this type of idea is much more salient to me than the collapse, the wave function collapse. So I'm actually like, if you know, if that's the case, then PF is now my you know, tied for my favorite quantum theory if it holds up. So yeah, I would definitely be stoked about that. Based on the provided text for presentist fragmentalism and standard scientific definitions of the many world interpretation, the two theories offer radically different pictures of conscious experience. They fundamentally contradict one another regarding the unicity of you, whether there is one of you or many, and the mechanism of your experience, whether you are creating reality or just sliding along a pre-existing track. Well, I think both would probably... okay, we'll see. The nature of conscious experience. Presentist fragmentalism. The actualizing consciousness. Conscious experience is defined by the A series, the dynamic flow of time. Experience is a filter of becoming. The text states that the A series flow represents the fundamental becoming of reality. Your consciousness operates at the leading edge of this flow where future possibilities actually... Okay. I take everything I just said. This disqualifies PF from my view, because it's missing the points of neuroscience. What neuroscience suggests about our conscious experience is that whatever reality we think that we're experiencing does not deserve the status of ontological truth. So it's not that, you know, when we talk about reality in terms of phenomenology, we can't say that that's some objective reality that's external from us. That is our experienced reality. It's what we feel in the moment, and it's dynamic. But yeah, if the A series flow represents this like this phenomenological aspect of our conscious experience, then this is looking in the wrong direction, I think. Your consciousness operates at the leading edge of this flow, where future possibilities actualize into present experiences before receding into the fixed past. Yeah, this is ridiculous. I take it back. I actually have to stop right now. I don't even want to entertain this anymore. Experience is singular but fragmented. You, Alice, my name is Alice, have your own independent now. You do not experience a superposition of outcomes. Instead, when you measure something, your timeline synchronizes with the object's timeline, and a single determinate property emerges in your shared present. You see that? This is like this idea of a determinate property being some property of the universe is what I am pushing back on. I think we know enough about neural correlates to know that our conceptual thoughts, our determinate properties, are manifestations of specific processes of the brain. And thus, we have a clearer view of how to make sense of a determinate property than this type of view. All right, we got to get out of here then. All right, did I ever go to this? Did I ever read this? I did not. This is the crucial realization. They are not two separate jobs. They are physically coupled. You are asking if the astrocyte has to choose between cleaning the mess and ordering the food. The answer is no. In the brain's brilliant design, cleaning the mess is the signal to order the food. Right. Okay. Astrocytes need the initial energy to clean the mess and signal for more food. Cleaning triggers feeding. So what happens if it's too much? When glutamate levels get dangerously high, the astrocyte doesn't stop fetching glucose. In fact, it is fetching glucose at maximum capacity. The problem is the bottleneck, right, the traffic jam. It's cleaning up as fast as it can and pulling glucose as fast as it can, but if the neuron keeps firing, the glutamate is entering the synapse faster than the astrocyte's pumps can remove it. Even though the machinery is working perfectly, the sink is overflowing, the unclean glutamate starts floating away and irritating neighboring neurons. Oh no! This is when the brain triggers the fatigue feeling we discussed. It forces you to stop adding water to the sinking ship. All right. I think we're going to chill before we get into oxytocin and opioids and social stuff, and we'll just call it a day. Sorry for going so hard on PF at the end there, but it has to be done on this channel. We gotta be stern about our ontological assertions, because that's what my understanding of the science forces me to do. You know, it's not because I just want things to be true. It's because I don't think the science allows us to think that way. All right. Let us call it a day. Hopefully I can internalize this stuff. Let me just try to look through at everything we talked about today. This is where we started. We talked about dopamine and opioids and endocannabinoids, reward prediction error. We talked a bunch about am I going to be able to find the start of this? No, that's not it. Oh, we started with this, actually. Yeah, long-term potentiation and prediction reward error. Reward prediction error. This had us thinking about how long-term potentiation works and what's happening with the chemicals that are tagging synapses for future protein strengthening and whatnot. And then we were talking about could we game that system. We talked about these two different types of dopamine, boosting signal and suppressing noise. Serotonin and satiety. Serotonin and dopamine are against each other. Gut-brain axis and serotonin and dopamine and all that stuff. Impulsivity in the weighting system. The effects of serotonin going down. All right, then we talked about glucose lactate and the astrocyte. That's the type of system we just talked about, where it's going to get glucose from the blood, it translates it into lactate for the neuron. The astrocyte's doing this work. Chill dude. I think we like all the glial cells on this channel. You could quote me on that. There's no glial cell we don't like here. Except that we do kind of like the microglias the most, or the microglia. Yeah, all right, we talked about glycogen, the reserve tank, glycogen being held by astrocytes. We talked about cortisol, how it gets the system going, but it also can start damaging neurons. We got adenosine. Oh, what was the thing that was causing... Oh, okay, yeah, the lack of dopamine can cause damage to synapses. We got adenosine. This is your cost of thinking throughout the day, your cost of just being awake, I imagine. You get it back when you sleep. Caffeine can make it have less of an effect for as long as the caffeine's working. But But also, I imagine adenosine is good for trying to fall asleep. Creates sleep pressure. Tuning the signal, GABA, glutamate. GABA inhibits, glutamate excites. There's a balance here. We got PV plus being the local metronome, and then we got the claustrum we were talking about, acting as the coordinator of these local metronomes, as well as I guess I guess the claustrum would determine which systems are talking to each other through this type of mechanism. That's what I'm guessing. Dysfunction, all right, glutamate accumulation. All right, then we got glutamate. This is accumulating when we're working hard in the moment. This can go away though if you give it time to rest, if you give the astrocytes time to clean up. So I think we're good. I think we're chilling. We talked about blood vessels. We talked about capillaries. Right? That's the word. Capillaries. Capillary. Yep. Density. Packed with capillaries. Density so high that no neuron is more than 20 microns away from a blood vessel. That's why I was looking at those blood vessel pictures, the capillary pictures, and I was thinking that it was not enough, because I imagine neurons are pretty. I imagine the brain's densely packed with neurons. Therefore, there's got to be a ton of just really small capillaries going everywhere. Like it would be a disgusting view, which is kind of what I was looking for. But all right, we're gonna continue tomorrow with this. I'm gonna try to like do some non-sleep deep rest right now to get all this stuff to stay. We're not gonna do this. I was gonna do something else today. I was gonna do this network planning, but we're not doing that. Instead, we did more deep dive, more brain deep diving. And tomorrow we're gonna do the usual taking recommendations. Though I might start off with like that Chris Williamson lessons from 2025 video, just because I'm curious what he's learning. Since I don't actually watch the podcast. And yeah, let's call it there. Thank you all for coming in and chilling. Thanks for saying hi. And oh, I got added. Oh, did I not respond to something? But what if presentist fragmentalism is discovered by scientists and philosophers to be effective at reconciling special relativity and quantum physics out of curiosity? I mean, that's fine. It's just that it has to compete against other system or other frameworks that reconcile special relativity and quantum physics, such as many worlds. And the issue is that it's operating on axioms that we know are inappropriate to operate on. So regardless of whether they can like fit it to test, like that's not to say that if they were to have tests that show some resonance with reconciling special relativity and quantum physics, then I would say let's not throw that out, but we need to throw out the way that they're like the conclusion that they're coming to, or the axiom that they're leveraging to get to that conclusion. Like we have a semantic issue here, where despite these tests that look good, you're operating on a faulty setup. Whereas even though, like, you know, I wouldn't say that Many Worlds is perfect, because Many Worlds is operating on this weird setup where you have many worlds, and we can't intuit that at all. That's an issue. But it's not as big of an issue as operating on something that we know is actually inappropriate, which would be to say that there is some reality because like affected by, or a manifestation of some external reality that's in the same domain as scientific truths when we experience the world. Like that would be inappropriate. And that's what I'm harping at. Like we have to keep, like when we're talking about reality, we have to keep conscious reality separate from physical reality. And if we're going to have a discussion about how we connect those, it has to be, as far as our science allows us, it has to be within the brain and neural correlates. Like, that's it. We can't jump the gun and start saying that because something is being experienced, therefore it has some ontological semantic authority in the physical world. That's the issue there. So yeah, I mean, that's not, yeah, not to say throw it all away. It's just like whatever thing is, whatever tests that they're using to reconcile with or reconcile special relativity and quantum physics, that they just yeah, they can keep that. They just need to figure out how to, linguistically drive this theory such that it actually works according to the norms of these other players involved. All right, I'm gonna call it there. Thank you though for the questions. Thank you for the paper, and thank you for getting me to see that stuff, because that's more stuff to learn, more cool stuff. It's regardless, I think it's good that I just have a context of what's out there, and yeah, this is all good exercises, good reps of thought. All right. Have a good rest of the day. See everyone later. Peace out.